cells are able to make, in some cases with other viruses, literally thousands of new viral particles within a single cell. They're able to do this by converting the metabolic machinery from the normal activity into the production of viruses. This involves the production of the new enzymes that we will be talking about today. They use the energy of the cell and the building blocks of the cell in the formation of these new viral particles. Now, outside the cells, viruses appear almost inert, and if you purify them and concentrate them, it's possible in some cases to obtain them in the form of crystals. For example, if you take a crystal of poliomyelitis virus, the first obtained in the virus laboratory in Berkeley, and slice through this crystal and look at it by means of the electron microscope, this is what you will see. Now, this has been enlarged about 1 million times. If you would take this picture that you see here and extend it for about 1 mile, the material that would represent in true life actually could be put through the eye of a very tiny needle. So this is a tremendous magnification. Now, if you take one of these little particles that you see here, which represents an individual polio particle, which, by the way, could cause disease in man or in animals, and enlarge it another 65 times, it's possible to get a model such as the one that you see here. Now, this model actually represents about all we know about poliomyelitis. Here we have a portion of a model of one of these viruses, tobacco mosaic virus. Actually, this is only about 1/10 of the entire structure of one of these viral particles. It's composed of these protein subunits on the exterior, and on the inside we have nucleic acid. Now, it's amazing that all of these diverse-shaped objects have the ability, when they get within certain living cells, to change over the entire inside, the metabolism of these cells, from making ordinary normal proteins and normal nucleic acids and make these virus structures that you have seen here. This is a perfectly amazing phenomenon. Just one of these particles going into the factory that we call a cell and changing the entire insides of that cell with the formation of new enzymes and new building blocks to make not normal things, but these viral particles. Now, this has been a particular area of interest to one of my colleagues, Arthur Pardee, and I'd like to have him come and tell you something more about this very fascinating subject.
Today, I'd like to talk to you a bit about how viruses manage to harm us. You know that viruses can do a great deal of damage. They can make us sick, they can paralyze us, or cause cancers, or kill us. If we knew something about how viruses manage to do these things, we would perhaps be able to prevent some of their actions. Now, the first thing that strikes us about a virus is that it's very small, much smaller than we. We can't even see a virus under a high-powered ordinary microscope. And you choose a simpler model, and it's a little embarrassing because it reminds one of the story of the fellow who lost his keys in the dark and then looked under the light post to find them because there was more light. We always try to look where there's more light also. So we do experiments with bacteria, sometimes called germs, which are themselves single cells of very small size. In the flasks I have here on the desk are some cultures of bacteria. The cloudiness is due to the bacteria. Now, I have 2 flasks here which are the same, and in here I have some virus. I'm going to add some virus to one of these flasks, and we'll see what happens. Now we'll take these flasks and put them someplace where they are able to let the bacteria grow, and we'll look at them after a little while. We know from electron microscope pictures, such as this one, that the viruses, the small dark objects in this picture, attach themselves to the bacterial cells. Then what happens can be summarized by the animated film that we will look at this point. In this film, we see a cell, and as we come closer to it, we see the wall of the cell, the outer barrier of the cell. And along comes a virus which attacks this bacterial cell. It attaches itself, and then the genetic material, the hereditary material of the virus, is injected into the bacterial cell. Most of the virus stays outside, but what we see is that the genetic material, the dark ribbon, enters the bacterial cell, and nothing seems to happen for some time. There is no virus inside this cell, no infectious virus, and many tests fail to show any change. But suddenly, the genetic material starts to duplicate, and many new models of this genetic material appear. And these are folded up inside new virus particles. Hundreds genetic material is built of chemical subunits, which are represented by the small white rectangles next to it. Now, we will see the things that happen in a bacterial cell before it is infected with virus. We see the food particles coming in from the soup in which the bacteria grow. This food particle is changed under the control of the genetic material called DNA into the different proteins and enzymes that are characteristic of the bacteria. This process goes on at incredible speed. Actually, billions of changes of this sort occur every minute in each tiny bacterial cell. These triangles represent special enzymes that are used in producing viruses. These enzymes are not found in the virus particles, but they are necessary for the production of the virus particles. And then we have the strip which represents the genetic material, the DNA of the virus, and we have the little rectangles which represent the parts of the DNA of the virus. Now, let us look at some more animated movies to see what happens in a virus-infected cell. Here's our bacterial cell growing and making its genetic material as it was before, taking in the food and changing it to the various bacterial parts. The viruses escape from the bacterial cell. They are free to infect other cells. In this way, a virus can create new viruses which can attack other cells, and eventually thousands and millions of cells can be killed due to the original infection by only one virus. Whole cultures of bacteria or whole groups of animal cells can be destroyed in this way. The cell, of course, is dead after the virus has attacked it. If we look at an electron microscope picture of a cell, a bacterial cell, after virus infection, we see something of this sort. The bacteria, the large oval objects, are flat and almost empty of their contents. We have new infectious virus particles in the bacteria, and at this point, the attack on the cell wall begins. The enzymes chew holes in the cell wall, and the viruses escape. So we have seen how one special bacterial virus manages to kill cells and to multiply. As I said earlier, this virus serves as a sort of model to help us understand how other viruses act. We know very little about the actions of many other viruses. We do know that they occur in many different shapes. For example, here is the sort of virus that we've been discussing, a typical bacterial virus. Here's a picture of some minute cancers that have been created by infection with a virus. Each of those little masses of cells show a place where the cells are growing faster than normal under the influence of virus infection. Still other viruses, for example, the pox virus kills cells in a local area and causes the appearance of these pustules, which eventually heal to leave deep scars if they don't kill the victim. You can see from a picture of this sort that viruses are nothing to take lightly. We then have the very practical problem of how to prevent virus diseases. And in particular, we'd like to know whether the kind of information that we've been discussing here will help us to cure virus diseases.